Hello everyone. I'm Dr. Shulagna Datta, Head of Service Research and Senior Faculty in Dentistry, Masa University. I would like to welcome all of you in a very special research sharing session today, which is a part of our faculty research clinic. And the topic is very interesting. It's about the p53, which is a very versatile molecule, and its relation in the childhood cancer. So, I would like to invite my friend Dr. Liu Fongfong to welcome and introduce our distinguished speaker from University of Malaya.
Well, good morning, Prof, doctors, my colleague and students. Okay. Right, thank you for participating our first research sections of this year. Okay. Today we have a very special guest speaker, Dr. Oli Zian, senior lecturers from Department of Pediatric, Faculty of Medicine, University of Malaya. Okay. Dr. O is a molecular biologist. Okay. She has double degree on the Bachelor of Science, Biotechnology, and Bachelor of Science, Medical Biosciences at Monash University. Okay. So late after she completed her degree, she furthered her study for PhD in University of Malaya. Okay. Currently, her research on DNA, RNA, protein, and cellular assay, bioinformatic analysis, public data mining, and statistical analysis to study the molecular mechanism underlying the pathogenesis of human diseases. Her current research work involves several clinical lab studies an international clinical trial in the area of childhood cancers, cancer survivorship, hematopoietic stem cell transplantations, and other pediatric disorders as well. Okay, extending from her PhD study, Dr. O explored the functional state of the tumor suppressor p53 in childhood cancer from the aspect of mutation and epigenetic regulations. Okay, I believe that this research work brings new viewpoint on p53 in childhood cancers and challenge the classical p53 concept, which was built upon the foundation of knowledge from adult cancers. Okay, so welcome our speaker of today, Dr. Oli Zian, for deliver a session on p53, guardian of the genome in childhood cancers. Thank you. Dr. Oli Zian, please.
Hi, a very good morning and Happy New Year to all of you. I hope you all can hear me clearly. So first of all, I would like to thank Masa as well as Dr. Liu Fongfong for these invitations, giving me this opportunity to share some of the information that I know about this tumor suppressor protein p53, and particularly in a childhood cancer settings, as well as to introduce to you all some of my research work. So I guess I'll try sharing my slide. Hold on a second. Can you all see my slides? I hope it's fine. Dr. Liu, can you all see my slides? Just to confirm.
Yes, we can see your slides.
All right, great. Okay, just to double confirm before we start. All right, so that's the title that we'll be looking into today, which is p53. Is it a guardian of the genome in childhood cancers? So again, I'm Lisian from Department of Pediatrics of University Malaya. Okay, so when we look into cancers, putting cancers aside, just looking into the normal cellular physiologies. Under the normal settings, our stem cells would divide, or we call it proliferate, and it differentiates into specific specialized cells to allow us as an organisms to grow as well as to repair any damaged tissues. However, in the event that the cells are damaged, that these cells should be directed to death via the process of apoptosis or go into senescence. So, but what happens if this normal cell cycle regulations goes haywire? Cancers may occur. So cancers is actually the disease when there is an abnormal or damaged cells starts to grow uncontrollably when they shouldn't. It goes just grow outside of the usual cell cycle regulations. And so what happens is that these cells will form into tumors, and it has the ability to spread as well as invade into nearby tissues, replacing the functional cells, interfering physically with the functions of the whole organs, as well as causing the imbalance in nutrients and so on. So just interestingly, I was just randomly Googling about those word cancer, which also means crab in Latin words, Latin language. So I came across this saying of these Greek mythologies that cancer is the astrological science known for their abilities to be in multiple realms. And it actually have a hard shell to protect themselves from the external forces as well as outside threats. So I was thinking, isn't this fits into the features of the cancers that we know, the disease cancers that we know today? It has the ability to spread from one side to another, as well as it always try to build up their own protective microenvironments which support its growth, as well as trying to evade from our human immunity that will clear them off. So when we ask, sometimes we ask layperson, or sometimes we even ask our doctors that, are cancers actually preventable? What are the risk factors for cancers? So you probably know some of the classic risk factors. One of it will be smoking. Your doctors will be telling you quit smoking, as smoking has been shown clinically associated to lung cancer's developments. And the others will be family histories, hereditary, or sometimes what we call the inheritance of a germline mutations, meaning that the mutations that we gotten from our parents, either of our parents. So one of the classic examples will be the mutations of BRCA1 genes that's associated with breast cancers. So probably you all have heard of how Angelina Jolie, she knows that she actually has a very strong family history of cancers, and she's suspecting she may have the BRCA1 mutations, germline mutations. Hence, she went for the mutation screenings, and yes, indeed, she has the mutations. So what she did is she actually go for a prophylactic mastectomy, meaning that she removed the breast tissues to prevent the occurrence of tumor developments. And the other thing is that although we know that cancers are not infectious, it is a non-communicable diseases, but cancer may be caused by infectious agents such as HPV, the human papilloma virus, which is associated with cervical cancers, or it can be the EBV, the Epstein-Barr virus, which is associated with nasopharyngeal cancers. And the other one of the risk factors that has been studied for cancers is radiations, like the UV radiation that has been associated with skin cancers, and that's what happened to Hugh Jackman. And he's always remind us about the importance of applying sunscreen. So these are the classic risk factors that has been studied and clinically associated to human cancers. But have you ever think that, how about childhood cancers? Isn't kids are just too young to be exposed to these lifestyle risk factors like carcinogens, like the cigarettes, the radiations, and so on for a long period of time? So all the risk factors that we discussed just now are actually mainly linked to the development of adult cancers. Whereas in childhood cancers, which we usually define it as those occurring in kids, in children between birth and the age of 15 years, these childhood cancers are actually not strongly linked to lifestyle or environmental risk factors. So a bit of statistics. Childhood cancers are actually the leading cause of death by disease in children. it is just the second following after accidents. And worldwide, every 3 minutes, a child may be diagnosed with cancers. And imagine, from the start of my talk until now, it's about 6 minutes. That's probably 2 children has been diagnosed with cancers. And how about in Malaysia? In the data gathered in year 2020, there are about 1,126 cases of childhood cancers diagnosed in Malaysia itself. So it's about 1 in 455 children diagnosed with childhood cancers before the age their 15th birthdays. So if you think that childhood diseases are just diseases in little adults, it is actually not true. Childhood cancers are actually biologically, histopathologically, clinically very distinct from adult cancers. So here I show a pie chart showing the type of cancers, the site of cancers that is present in adults, the pie chart on the left, as well as in children, the pie chart on the right. So here you can see that the presentation of the cancers among these 2 age groups are actually very different. Adult cancers, the most common one mainly are, as you can see here, will be lung, breast cancers, colon cancers, rectal cancers, prostate cancers. And all these cancers actually arise from mature epithelial cells derived from the endoderm and the ectoderm region of our human organisms, giving rise to carcinomas in these organ sites. Whereas for childhood cancers, the most common type of cancers are leukemias, or cancers of the brain or central nervous systems, as well as the blastomas, the sarcomas, that is derived actually from immature cells that is in the early stages of development. So knowing that childhood cancers actually behave or present in a very different way as compared to adult cancers, as well as their associated risks. To date, causes as well as associated risks for childhood cancers are still poorly understood. It is, just now as we mentioned, it is rarely associated with environmental factors such as infections, radiations, pollutants, and so on, as what we usually see in adult cancers. Whereas in child cancers, to our surprise, it's only 6% is associated with family history, or what we call germline mutations. And whereas most of the childhood tumors, childhood cancers, actually occur sporadically in families with very weak family history, weak inheritance history, then the causes of all these, majority of these childhood cancers still remains unknown. So as we mentioned just now, cancer is actually a disease of uncontrolled cellular divisions or proliferations. So one of the postulations for the etiology of childhood cancers is that rather than it being the direct results of oncogenic transformations by environmental carcinogens and so on, the formation of childhood cancers, could it be actually driven by the defect in the growth and differentiations promoting mechanisms during organ development, meaning that there's the balance between proliferations as well as differentiations has go haywire. So from the proliferations, plasticity, as well as invasiveness features that we observe in childhood cancerous cells actually reflects that the failure of all these immature cells to undergo normal maturations during early development. So there is actually a lot more about childhood cancers that which we need to understand, which we need to look into. And indeed, fundamental scientific research in childhood cancers has been quite limited, especially in Malaysia. Okay. So looking into how about, just now we're looking into statistics in terms of incidence, prevalence, how about its curing? So with the advancements of all these therapies, such as the conventional therapies like cytotoxics, chemotherapies, radiotherapies, in conjunction with surgical resections, where it's appropriate, more into solid tumors. So the overall cure rates for childhood cancers have increased to 80%, in most developed countries. That's provided that these children actually get diagnosed and get access to medical treatments. So that's like the average cure rates. However, because all these chemotherapies, irradiations, they generally just target rapidly dividing cells. And as such, it may concomitantly damage the development of these organs in all these affected young children. And that's why, even though they actually survive through this cancer disease, when we follow them up after their completion of their treatments, when we follow them up years later, we realize that 2/3 of all these survivors, they actually experience late effects. So they experience late effects such as organ toxicities, like heart damage, lung damage, and so on, cognitive disability, impaired cognitive abilities, as well as the much higher risk for secondary malignancies. That's why now we say that there is actually a need for a more specific and a less nauseous therapies to manage cancers in this young children's group. And the first step for this strategy is actually for us to have a better understanding of the biology of childhood cancers, which now we know that is actually very different from adult cancers. So if you recall just now, I highlighted that about 6% of childhood cancers are actually associated with family history, with hereditary predispositions or germline mutations. So a bit of history. When I first joined the oncology research teams years ago, the first case that I was introduced to was actually Lee-Fromentin syndrome, or we call it LFS in short. So this LFS is actually a hereditary cancer predispositions syndromes associated with a germline mutations in p53. So this LFS is actually a devastating syndromes that actually predispose individuals at a very high risk for a diverse spectrums of cancers. And the lifetime risk of cancers in these individuals is about 80% for men, meaning that if you're a man, if the LFS individual is a man, there is an 80% risk for him to actually get cancers throughout his life. And for women, it's actually slightly higher, it's 85% because of the development of breast cancers. And if you are familiar with like inheritance pedigrees and inheritance patterns, this LFS is actually a genetic disease that is inherited in an autosomal dominant manner. So what exactly is LFS? So the diagnosis of LFS is actually very clinical based and actually requires extensive analysis of family histories. So one of the most widely criteria that we use is actually the Schomburg's criteria, as I shown in the table here. So what happens is that when the clinicians see a case with a tumor belonging to a specific spectrum, we call it LFS tumor spectrums, and it actually have not just that, it have at least one first or second degree relatives with an LFS tumor as well, before the age of 56 years old, means at a much earlier, younger presentations of cancers, then clinicians may be suspecting LFS for these individuals, for these children, and the clinicians will advise the kids to actually undergo germline mutation screenings for p53. And that's just one of the criteria, but there's also another scenarios whereby if a kid is presented with multiple tumors, that is belonging to two of it belonging to LFS spectrums, and all these tumors actually occur before the age of 45, that is also suspecting for LFS. So why is it important for us to actually identify whether this patient is LFS individuals or whether this patient is actually having a germline p53 mutations? Why is it important? So of course, one thing is that we can then perform genetic counseling for the family if they are planning for another kid. But not only that, it actually have an implications in terms of the disease treatments. So clinical studies, as well as mouse animal models, as well as cellular in vitro studies, have shown that individuals carrying germline p53 mutations, radiotherapy, as well as all these conventional chemotherapies, will actually lead to the development of the subsequent primary tumors. They are at a much higher risk for that. That's why for LFS individuals, the priority should be given that they should be given surgical or ablative treatments. Try to avoid, if possible, any genotoxic treatments for them. Not only that, there's also surveillance recommendations for these individuals whereby they'll be recommended to perform clinical examinations as well as scannings throughout consistently throughout the period of time that is recommended during the time frames. So like for example, MRI, brain and whole body should be done annually and so forth. Just try to detect if there is any early signs of cancers. Okay, so that's about LFS. And actually years ago, just showing some of the research work that my group and we actually did, is that years ago we were actually interested because all the knowledge and information that we know about LFS are actually coming from the European or the American cohort. So then we are interested to see if the LFS families, LFS individuals in our Asian cohort, do they behave similarly as our Caucasian counterparts? So what we did is that we dig out all the germline p53 mutations data from this comprehensive IARC-TP53 database that's actually maintained under IARC, which is currently under the National Cancer Institute, NCI of United States. So what we did is that with that information, we actually tried to plot the cancer rates, or what we call the penetrance rate for these individuals, for these LFS individuals. We split it into 3 groups based on the geographical regions: Asian, the North American, as well as the Western Europe. So here we see that before the age of 35, about 59% of these LFS individuals were presented with cancers. It is actually more than half. So imagine before the age of 35, more than half of them will actually be presented with at least one cancer. But beyond the age of 35, you can start to see the divergence in the Asian cohort, which is shown in the green line. You see that the penetrance rate is actually different from the North American and the Western Europe. By the age of 50, you can see that up to 90% of the Asian p53 mutations carrier will actually develop cancers. Whereas for the Americans as well as the Europeans, their cancer risk is just about 80 to 82%. So then we try to investigate what could be the reason behind this difference that we observe between the age of 35 and 50 that's different in our Asian cohorts as compared to others. So what we did is that we examined the type of tumors that we observe in these 3 geographical cohorts. And we find out that stomach cancer, the gastric cancers, was the only tumors that show differences across these 3 regions cohort, whereby it's about, in Asian cohorts, 16% of the cancers that is observed are stomach cancers, whereas it's only 1%, it only account for about 1% or to 4% in the European cohort. So some may be saying that, well, yeah, because gastric cancers is a very prevalent cancers in the Southeast Asia. So what we did is we adjusted to the population risk, and yet we still find that the incidence of gastric cancers in Li-Fraumeni syndromes individuals are still much higher in the Asian cohort as compared to Caucasian cohort. So this actually tells us that the higher incidence of gastric cancers in Asian p53 mutations carriers is not just solely attributed by the population's risk that we usually see higher in Asia. So what does this information actually brings about? With these information, we propose to this Li-Fraumeni syndrome consortium that they should actually consider adding the early presentations of gastric cancers, and we say early as in diagnosed before the age of 40, into this modified Tumor criteria, especially for the Asian populations. So here I just demonstrate one of the examples as how we actually use publicly accessible comprehensive databases that's available out there to answer some of the research questions that we have. And using this finding, and we actually translate it into a clinically important information that can be practiced in patient settings. Okay. So that's about Li-Fraumeni syndromes, which is a classic examples of hereditary cancer predisposition disease that we see in children. So, and just as we mentioned just now, hereditary is only associated to about 6% of childhood cancers. So how about the rest, the majority of it, the 80 to 90% of childhood cancers, which actually occur sporadically without family histories? So maybe some of you are familiar with this. It's called the Hallmark of Cancers, which is by Hanahan and Weinberg, that they actually summarize the principles or the features of cancers, which includes like sustained cellular growth, resistance to death, as well as evading immune destructions that we mentioned just now. So interestingly, when we actually look at this hallmark of cancers, you will realize that there's actually one protein, p53, that actually just fit nicely into these circles, in which that this p53 protein, the tumor suppressor protein, it has been shown, multiple studies have shown that it actually able to interfere with each of these 10 hallmark of cancers. So this p53 protein, that most of y'all know it as a tumor suppressor in the context of cancer field. But actually now with the extensive research in various fields, researchers have found that actually p53 is not just playing a role in cancers, but it actually also have a lot of physiological roles in development, in differentiations, in immunity, cellular metabolisms, and so forth. And that earning it the name of guardian of the genome. So what exactly does this p53 protein does? So this p53 protein, it actually functions as a tetramer, meaning that it actually needs 4 subunits to be a functional protein. So it functions as a tetramer, and it acts as a transcription factor. So it earning the name of guardian of genomes, what it does is actually it responds to a variety of stress-inducing signals. So for example, DNA damage, hypoxia, telomere shortening, as well as oncogene activations. So when it actually encounters these stress-inducing signals, it will respond to it and it will get activated. When it gets activated as a transcription factor, it actually able to activate the expression of a lot of its downstream genes, or what we call target genes. So here I put a few examples, such as the CDKN1A, 1433 sigma, as well as BEX, and so forth, GADD45. These are the p53 target genes that's involved in a range of biological outputs, such as cell cycle arrest, senescence, apoptosis, DNA repairs, differentiations, and so on. So all these biological reactions are actually in the directions to limit the growth of a damaged or a potentially neoplastic cells. So hence, trying to prevent the growth of the development of cancers. So this p53 protein activities is actually indeed controlled by many ways. So here I show a few of the classical regulators. Here, one of it will be the negative regulators, MDM2, whereby when MDM2 actually binds to this p53, it will direct it to the proteasome mediated degradations, meaning that the p53 will be degraded and hence not functioning. The other negative regulators will be MDM4. Besides that, p53 can also be regulated at a post-translational stage via post-translational modifications such as phosphorylations, acetylations, methylations, and so forth. And the activity can also be affected by its isoforms, which is actually the shorter form of the p53, which we will go further into later on. So actually, since the discovery of this p53 back in 1979, so it is about 43 years by now, it is actually one of the most investigated molecules ever. It is the subject of more than 100 over thousands research papers. However, despite the attention, so much attention given to it, much remains to be understood about these proteins, about how p53 as a single protein is able to regulate such an extensive network of biological processes, and how this p53 actually being regulated in different cancer settings or different disease settings. There are still a lot more knowledge gaps remains to be explored. So what actually makes p53 one of the most famous protein is that it is indeed the most mutated genes in adult cancers. As you can see here, I show that TP53, which is actually the gene coding for p53 proteins. So it is the genes that is being mutated somatically, means not inherited from parents, but it's just mutated in the cancer tissue cells itself. It is observed in close to 50% of adult cancers. And all these cancers with the p53, with the mutated p53, they are usually associated with very poor prognosis, resistance to chemotherapies, and so on. So that's about adult cancers. So how about childhood cancers? So in general, mutations loads are very low in pediatric cancers, in childhood cancers, as compared to adult cancers. However, as you can see in the figures on the right, when we try to rank the genes based on the mutations rates, TP53, which is the gene coding for p53, it still remains as the top mutated genes among childhood cancers. But it is only present in about close to 4% of child cancers, as compared to adult cancers where you can see about almost 50% of them are with the p53 mutations. So it is actually intriguing that given the significance of p53 in tumor development, as we see just now, these inactivating p53 mutations are so rare in child cancers. So this phenomena actually raised my research question as to figure out why is there a low mutations of such an important protein in child cancers? I wonder how these cancer cells without p53 mutations able to survive through this carcinogenesis, even if the presence of a possible p53 tumor suppression. And I wonder what exactly is the role of this protein in childhood cancers. So here we come out with some hypothesis is that since majority of the childhood cancers may retain an intact wild-type p53 genes, what happens is that in child cancers, it may actually activate alternative mechanisms that would actually inactivate the p53 functions. So when we say alternative mechanisms, some of the examples will be upregulation or high level of the negative regulators, the MDM2, MDM4 that we mentioned just now, or it can be a high level of the isoforms that react in a dominant negative manners that inhibits the functional full-length p53. So here is just relating back how the hypothesis about the development of childhood cancers as well as the role of p53 in differentiations. So here I present one of our hypotheses is that during the early embryonal developments, during the early organ developments and so on, these p53 proteins remains predominantly in an inactive state. However, when there is a differentiation induction in differentiation signals, what happens is that this p53 will be activated and stabilized. And it drives the differentiation of all these stem cells to a specialized committed cells or differentiated cells, and that eventually develop into the respective organs. So what happens if this p53 is not functioning? When it's not functioning or inactive, we say, these stem cells may retain in its pluripotency states, in the proliferation states, and it may accumulate genetic damage caused by errors in replications, in DNA repairs, and eventually leads to the development of childhood cancers. So as majority of, just now we mentioned, the majority of childhood cancers actually have a wild type p53. And we postulate that this p53 activity, it can be functionally silenced through alternative mechanisms like disruptions of other members in the pathway, like MDM2, MDM4, or isoforms and so on. So if we can identify these alternative mechanisms and we reverse it and be able to reactivate back the p53, we may drive the cells into cellular differentiations and apoptosis, reducing and eventually driving the cells back into a differentiation pathways and reducing the tumor growth. And this will be actually a more promising and less nauseous approach in managing childhood tumors, where arrested organ development is predicted to be the underlying pathology. So actually, we published a review papers whereby we actually summarize our current knowledge about how p53 pathway could be inactivated in childhood cancers, which carries a wild type p53, meaning that they do not have the mutations, but what are the other mechanisms that would inactivate, would disrupt the activity of p53. So here I'll just simplify, I'll just put it a diagram in a very simplistic way. Of course, the regulation of p53 will be much sophisticated than what is shown over here. So here we have the p53, and we mentioned about the negative regulators, upstream regulators, the MDM2 and MDM4. So what happens if there is a high level of this MDM2 and MDM4, it will actually attach to the p53 and bring it to be degraded. And indeed, multiple studies have shown that these MDM2 gene amplifications, as well as the upregulated high expressions, has been shown in multiple child cancers like retinoblastomas, neuroblastomas, brain tumors, as well as lymphomas, in a subset of all these cancers. And those cases with this MDM2 overexpressions tends to be associated with a more progressive or a relapsed form of the cancers. And upstream of this MDM2, there's actually another molecule called p14R. So this p14R actually it inhibits the activity of MDM2. So imagine if this p14R functions is lost, it will actually enhance the activity of this MDM2 and causing the p53 activity to be low. So what we see is that this p14R inactivations is indeed also being demonstrated in a few subsets of child cancers, like neuroblastomas, retinoblastomas, as well as some of the hematological malignancies. So there are a lot more, but I'm just showing a few examples over here. So another examples of p53 regulations will be via the epigenetic manner, which is through microRNA. So these are the two classic microRNAs that will actually directly bind to p53 mRNA that stop the translations of this mRNA into a protein. So indeed, studies also shown that there is a high expression of such negative microRNAs present in medulloblastomas as well as leukemias. And these are actually similarly associated with very poor disease outcome. And another interesting bit will be the isoforms. Like we mentioned just now, is actually the shorter form of p53 that we will discuss further later on. It's just that there are so far, this field of p53 isoforms are still fairly new. And to our knowledge, in terms of childhood cancers, there's only very few papers looking into it. That one of it is that they detected isoforms whereby it actually been cut off or truncated in the C-terminus, and these isoforms has been shown in the neuroblastomas. Whereas in other cancers, isoforms has not been studied, but there are evidence whereby there are a splicing dysregulations present in Wilms tumors and leukemias and so on. And these splicing dysregulations actually give us some clues that could imply that p53 isoforms may be playing a role in these cancers instead because of these splicing problems. So what happens is that if this p53 is being inactivated via all these dysregulation in the upstream regulators instead of mutations, what happens is that these stem cells, which could already accumulated some of the genetic defects, they will remain in their pluripotency states, and they will continue growing and forming tumors instead of going into a differentiation. So that's about childhood cancers in general. So in an experimental approach, me and my team, we try to use the most common childhood cancers, which is the acute lymphoblastic leukemia, ALL in short we call it, as our primary study model. So we combine both experimental as well as bioinformatics and data mining approach. We try to investigate, we explore what is the p53 activation or functional status in these childhood cancers, the ALL. So I use original RNA-seq transcriptomic data sets of comprehensive cohort made up of about 200 cases, 200 leukemia cases. So what I did is that I tried to deduce the p53 activity states based on the expressions of its target genes. So from here, I actually plot based on the expressions of its target genes, the downstream target genes that's involved in multiple biological processes. Based on these expressions, I plot these PCA plots. And here I can see that those cases which is denoted with red dots is labeled controls. They are actually the healthy marrows, bone marrows taken from healthy children. You can see that all these healthy marrows actually cluster at one side. Whereas for those blue dots, they are actually the ALL cases, the leukemia cases, which is carrying a wild type p53, meaning that they do not have the mutations. And you can see that they are actually very spread out instead of they cluster just like the control samples. This actually signifies that there is a high heterogeneity of cases that is carrying a wild type p53 in terms of the p53 pathways. So what I did next is that I used isopods clusterings, and I tried to cluster all these p53 wild type ALL cases, and I clustered them into 4 groups, as shown by these 4 different colors over here. I name it as K1, K2, K3, and K4. So then I actually try to deduce the p53 activity states or activity scores for each of the cases in each of these K1, K2, K3, and K4 groups. So the algorithm of this calculation of p53 activity is actually more complex, but I just put it in a very simple manner is that I measure the activity, I deduce the activity of p53 as a transcription factors based on the expressions of its downstream genes. So if let's say I'm looking into 16 downstream genes, if in this leukemia case, all 16 genes are upregulated, and I will give it a score of 16 out of 16. If there's another cases where I look into all these 16 genes, only 8 of them is being upregulated, then I'll give it a score of 8 out of 16. So that's just a simple calculation of that. So from there, I plot the p53 activity scores, and I realized that there's this one group called K4. It actually have a much significantly lower p53 activation scores as compared to the other three groups, K1 to K3. And the scores in these K4 groups is actually somewhat similar to all the cases which is carrying a mutant p53. So, and then interestingly, when I actually correlate to the clinical prognosis, clinical data, I found out that these K4 subgroups appear to have a marginally more inferior event-free survival outcomes. The outcomes is about 85% as compared to the other groups, which is close to 90 to 95%. So this suggests that although right now it's not meeting a statistical significance yet, but these observations actually does ignite or suggest to us that the differences in the degree of p53 activity may actually account for some of the clinical disparities in leukemias. So of course, these findings will require more verifications before we can proceed to the next steps. So following on that, then we would like to understand what actually causes, what are the mechanisms that is maybe contributing to this p53 inactivity in leukemias, in acute lymphoblastic leukemias. So from previous years, what researchers understand is that p53 is a unique molecule. But actually that's not true. There are actually many forms of p53 products and what we call isoforms. So I would like to pay credits to Prof. Pierre for these slides, for these interesting slides. So what exactly are isoforms? They are actually the clip form, or we call it the shorter form of p53. So this is the list of these p53 isoforms. So you can see that it's actually a truncated form. It can be truncated at the C terminal, which is here on the right side, or it can be truncated at the N terminal, which is on the N side. So these isoforms are actually produced via various mechanisms. It can be through usage of alternative promoters, as here we can see there is actually a P2, another promoters, or it can be a usage of a different translation initiation sites, or we call it the ATG, a different start site. Or it can be alternative splicings. Or it can be combinations of all these mechanisms that are giving rise to all these different forms of isoforms, which is the shorter form. Whereas the full-length p53, which is the functional form, we call it TAP53 alpha. So all these isoforms, the shorter versions, because they are lacking of some parts of the functional domains, these isoforms, are not able to behave like the functional full-length p53, the TA53. But what happened is that they could even inhibit the activity of this full-length p53 in a dominant negative manners. So instead of them, like the usual p53 that's supposed to act as a tumor suppressor, what happened is that all these isoforms, they may instead act as an oncogene instead. So what I'm interested is that I'll try to look into the expressions of all these p53 isoforms in a leukemia, in acute lymphoblastic leukemia. So I've looked into it, the expressions in normal marrows, meaning marrows from healthy children, as well as primary acute lymphoblastic leukemia, and a few cases of relapsed leukemia. And here I found out that in comparisons to a normal bone marrows, healthy bone marrows, what happened is that the full-length functional TAP53 alpha, they are actually overexpressed in a primary ALL. But in contrast, in the relapsed cases, there are actually upregulations. There is a high expression of all these alternative delta and p53 isoforms, which are the shorter form of p53 that is truncated at the N terminal. We call it delta N p53. So since just now we already mentioned that p53 actually works as a tetramer, so it needs 4 subunits. So I then use mathematical modeling to try to predict how the overexpressions of all these p53 isoforms could actually affect the compositions of all these tetramer complex and hence postulate the effect on the p53 functions. So in primary ALL, p53 is predominant, based on this mathematical model, we found out that p53 is predominantly exists in a homotetramer full-length p53 conformations, which is all with the blue subunits. Whereas in relapse cases, they are actually appear in a heterotetramer conformations, in which they have at least 2 or 3 delta N subunits, which is the conformations with a bit of orange and a bit of blue. So this actually results suggest that while primary ALL retains a normal pattern of the functional full-length p53, in relapse, the expressions of these dominant negative isoforms, the delta N isoforms, may actually dysregulate the p53 pathway in the relapse cases and contribute to a more aggressive behavior in a relapse diseases that we see. So in summary, our work actually brings about a new viewpoint about what is the p53 functional states in childhood cancers. It further established that actually in childhood cancers, there is a low selective pressures for a loss of p53 functions by mutations. Because these p53 functions, it could actually be dysregulated via other pathway. So one of the pathway that we've shown is actually the overexpressions of the p53 isoforms, the delta N dominant negative isoforms that we observe that may be inactivating the p53 activity in a relapsed ALL. Whereas in the primary ALL, as shown in our RNA-seq data as well as the isoform studies, is that they are mainly present in an active form, except for some small subset of it that seems to have a suboptimal activities. So of course, we know that all these works are actually requires more verifications and so on. And we are fortunate enough to actually secure a few grants to allow us to do more verifications and extensions of works, whereby we will validate our findings that we observed previously using a different cohort, as well as not just looking into isoforms. We try to explore, is there any other alternative mechanisms that may inactivate p53 in ALL, as well as extend it to other forms of child cancers. And just now we mentioned that we've observed that p53 isoforms may play a role in relapsed ALL particularly, and we would like to evaluate and find it out whether, if so, can we manipulate this p53 isoforms level, and manipulation of this, would it enhance the anti-tumorigenic response, and would it help and be able to incorporate it in as part of the treatment regimen. So again, same thing I'd just like to highlight here is that similar to clinical treatments, there's no one-size-fits-all kind of thing in treatment of diseases. And same goes with research. So here I'd like to highlight is that we may actually be using different approach. We may look into a same topic from a different angle, same research questions, but we actually explore it with a multiple different angles using different approach, different techniques. So here I try to demonstrate that we use public epidemiological studies, epidemiological database, as well as transcriptomic database, either it's publicly available or our in-house database, or we can use experimental work, resources from clinical trials, as well as cell lines models to try to investigate one of the research questions from multiple angles, so that with the aim to actually bring our findings from cellular models and so on to the clinical implications, to bring it from bench to bedside, to have implications in terms of treatments or patient's diagnosis. So some of the clinical implications of our work is that because we know that p53 mutations are actually very rare in child cancers. So meaning that therapies, a lot of therapies that are targeting mutant p53 would be of little use in childhood cancers, as majority of them are actually having a wild type p53. And if we could identify the alternative mechanisms that is inactivating the p53 pathway, then it will be interesting to test whether if we can reverse these dysregulations, are we able to activate back the p53 pathways, and would that actually enhance the p53 dependent suppressions and improve treatment response? And just like the work extension from the RNA-seq data that we show, we're able to identify a cluster, the K4, which seems to have a poorer disease prognosis. So if we can escalate that and establish this detailed analysis of this p53 transcriptome, then it will actually have a prognostic value for us to identify which are the leukemia cases that may have a higher tendency to be resistant to treatments or a higher risk of getting into relapse later on. If we can identify them, then it will be interesting, or it will be worthwhile for us to actually escalate the treatment protocols for them, so that we can escalate them to a higher risk groups, so that we don't undertreat them. So here I'll just like to highlight, or just to show to you all that just now I mentioned p53 are not just playing a role in cancer, they are also have multiple roles in other physiological processes like metabolic pathways involved in diabetes, lipid metabolisms, as well as immunity or some neurological diseases and so on. So you all can, if you're interested, you can actually explore into it. And as I understand, probably some of y'all may be of a dentistry backgrounds. So on a quick search, there are also a lot of people looking into p53 in oral care, like the prognostic value of the p53 expressions in oral squamous cell carcinoma, as well as they're looking into how p53 pathway may be dysregulated, whether is it by MDM2 in the head and neck cancers. So if anyone later on you may be interested to look further into these proteins, or if you are interested to discuss further, you can just contact me via emails and so on, and we can discuss through. So this, I just want to introduce, this is actually a book. It's actually a book, like a storybook kind of thing, that is talking about history and how p53 research comes by. It is actually dedicated to layman person. So it doesn't necessarily that only people with strong scientific backgrounds are able to understand. It's for layman person. So you can get it from MPH and I'm sure like other bookstore as well. So it's quite an interesting books that you can actually look into to understand this protein further. So with that, lastly, I would like to take this opportunity to express my gratitude to both my advisors and mentor, Prof. Pierre Henu, as well as Prof. Hani Arifin for their guidance and motivation throughout these years. So, and also I would like to thank my team members from the University of Malaya, collaborator sites, IABE in Grenoble, France, as well as NUS Singapore, with whom I actually work closely with throughout my studies. Right. With that, thank you for your attention.
Okay, thank you, Dr. Lizian. Okay, for this interesting and very informative sharing sessions. I think I received some question, okay, regarding related to today's sharing sessions. From Dr. Pallav, he mentioned that, okay, as among children, most prevalent cancer is leukemia, followed by others like CNS tumor, lymphomas, etc. How the p53 defensive mechanism or expression varies among these various cancers? And second question from him is there any correlation between the childhood obesity and risk of a childhood cancer?
Okay. Thank you for that. Thank you, Fong Fong, and thank you, Dr. Pala. Is that, as far as I understand, so the question is looking into how p53 mechanisms differ from brain tumors as well as leukemias. So one thing I would like to highlight is that when we look into brain tumors, there are many subtypes of brain tumors that we see in child cancers. There are some subtypes that actually they have a high mutations rate of p53. So if we disregard those, we realize that similarly as leukemias, that except for these particular portions of brain tumors, most of the brain tumors also carries a wild-type p53 as similar as leukemias. And we found out that actually a lot of studies are saying that p53, the regulations of it, whether is it by isoforms or by other NDM2 and so on, the regulations are actually very specific to tissue origins. So this is also something that we're trying to look into it. We realize that p53 behaviors, it may actually be different based on the embryonic tissue of origins. So we would say that, of course, at this moment of time, we have not looked much into brain tumors, but we will foresee that it could be, I don't know, it could be either in a similar pathway. So like just now we see there are certain of them still showing MDM2 amplifications in certain gliomas, as well as loss of p14F in certain brain tumors as well. But it will always be worthwhile to look into an extensive how p53 is being regulated in a disease-specific manner. So not just in leukemias, our understanding in leukemias may or may not extend to the brain tumor settings. Yeah, I hope I understand at least partially on that. And next will be the correlations into obesity and childhood cancers. To be honest, I so far I have not came across too much about this kind of correlations in terms of epidemiology or clinical studies. Yeah, so and maybe I can look into it and I can further will be able to answer this more confidently by then. Yeah.
Okay,
at this
thank you,
moment of
Dr.
time, I'm not sure of any significant correlations on that.
Okay, thank you. I think I have one question. I think throughout your presentation, I heard about p53 and TP53. So I think audience may wish to know what is the difference between p53 and TP53 as well.
All right. So p53 is actually the protein. So we call the protein 53 the tumor suppressors. And TP53 is actually the name, the gene name that is coding for this 53 protein. So protein level, you call it p53. At gene level, you call it TP53. So yeah, some people may be a bit confused on that. Yeah.
Okay, thank you. And then one last question for this session is, okay, I think some pathologies may know about p63 and p73 as well. Okay, so just wondering what are p63 and p73 in relation to this p53?
Okay, that's a very good question. So p63 and p73 is actually a family. p53, p63, and p73, they are actually a family. They are members from the big p53 family. So it's something like a siblings kind of thing. So they are actually sharing a lot of similarities in terms of structural. So, and studies have shown that because of this high degree of structural similarities, p63 and p73, they show that they are able to bind and activate some of the expressions of p53 downstream genes. So, but when they look into mouse knockout studies, so when we knock out p53, we realize that the mouse will have a high tendency of developing cancers. But then interestingly, when they have like p63 or p73 mouse knockout studies, they show that these knockout mouse, they actually do not express increased cancer susceptibility, but they exhibit severe developmental abnormalities, like the abnormalities in their CNS developments and so forth. And also, if you look into like epidemiology, you will realize that in human cancers, p53 is one of the most prominent mutations, most mutated genes. Whereas for p63 and p73, they are rarely the targets for inactivating mutations in human cancers. So, of course, there are also other studies like p73. So, same thing as p53, 63 and p73, they also have their isoforms. Then their isoforms, some of them they study also acting in a dominant negative manner. And one of a few studies that have, if I'm not mistaken, they have shown that dominant negative p53 isoforms, they do have some oncogenic roles in some of the cancers, but these findings are yet to be confirmed. So actually, it's a very interesting studies, interesting questions, because as we know, just now we mentioned p63 and p73, they are actually involved in developmental and differentiation. And one of our hypotheses of the etiology for human childhood cancers is that it actually relates to perturbations during differentiations and development. So actually, it's also part of our interest to not just looking into p53 pathway, but maybe later on we can also look into the interactions with p63 and p73 pathways. Yeah, so that would be the further works.
Okay, well, thank you. And I think after this session is very inspiring and very informative, and we all will get very important hinting on how to get more research on P53, 63, and 73. Okay, and we hope for a potential collaboration with you in future as well.
Yeah.
Okay,
Yes,
thank
definitely
you.
looking forward to that. Right, thank you so much again for your attention and this opportunity to talk.
Thank you very much, Dr. Li Xiang. Thank you very much, Dr. Fong Fong. I'd like to thank you, Dr. Xian, because it's very updated information on how modulation of P53 may ameliorate childhood cancer. And with this, I would like to add that even our respected research director, Prof. Shri, has commented that thank you for the very good talk, so clearly and succinctly explained. So thank you very much, and we would look forward to meeting you in person. Thank you.
Thank you. Thank you. Have a great day ahead.
Thank you so much. Thank you, all the viewers. Bye bye.
Okay, thank you. Bye bye.